For those who don't know me, my name is Megan Chambers, and I am the NIJ Program Manager for the Seeds, Drugs, and Toxicology Research and Development Profile, and I'll be moderating this session. So we have a wide variety of presentations and speakers that we have for you today. We have people from universities, we have federal partners, and we also have publicly funded forensic laboratories. So with that, our first speaker is Alexandra, or Alex Bryant. She is a PhD candidate at North Carolina State University. Her current project involves aptamer characterization using fluorescence-based assays for the identification of high-quality aptamers for use in the development of electrochemical aptamer-based sensors for the detection of illicit drugs. I have had the opportunity to hear a little bit about Alex's research recently, so I'm excited to have her present it all for you. So go ahead, Alex.
Good afternoon. Sorry. Okay, today I'm going to be sharing my presentation about identifying high-quality aptamers for drug detection. The ability to detect small molecules of interest is important for a variety of fields, from healthcare to food safety purposes and to forensics. And one way we can detect for these small molecules is through the use of instrumentation, such as HPLC or mass spectrometry. And though these instruments are very accurate and sensitive, they have some disadvantages in that they require highly trained personnel and they can be expensive to maintain. An alternative way to detect for small molecules is through the use of a biosensor. Two examples of biosensors that we're familiar with is the pregnancy test and the glucose meter. These are successful because they're fast, economical, and user-friendly. And one of the components in a biosensor is a bioreceptor. So this is what recognizes our analyte of interest. For a pregnancy test, it utilizes an antibody, and for the glucose meter, it utilizes enzymes. And though these biosensors have had their success with these bioreceptors, they do have some disadvantages that we need to address. One is that they have a limited analyte scope, poor stability, high batch-to-batch variation, and they're expensive. So now I would like to introduce to you all a new bioreceptor called an aptamer. So aptamers are single-stranded RNA or DNA oligonucleotides that can bind to specific molecules with high affinity. And they recognize their target through various interactions, such as hydrogen bonding, electrostatic interaction, and London dispersion forces. We isolate these aptamers on our benchtop through a process known as the systematic evolution of ligands by exponential enrichment. This process is also called SELECTS. I now want to give a brief overview of how the SELECTS process works. So we start off with a randomized library of 10 to the 14 unique sequences. We then take this library and we incubate it with a target of interest, for example, THC. We then are going to separate the non-binding aptamers from the ones that bind. The aptamers that do bind, we're going to amplify with PCR, and we're going to repeat this process until we have an enriched pool. From there, we can send out our pool for sequencing to identify the composition of these aptamers. Now, we start with 10 to the 14 unique sequences, but we end up with about 1,000 to 10,000. And from there, we need to find an aptamer that has good affinity and good specificity. And so how do we do that? Well, we have some gold standard methods, such as the isothermal titration calorimetry, microscale thermophoresis, and the surface plasmon resonance. These instruments can provide thermodynamic data, kinetic data, so they're very useful. But the problem is that they're low throughput, meaning I can only test one aptamer and one target pair at a time. And when you have thousands of aptamers that need to be evaluated, this can increase the time. Just to show a visualization, if you have 30 aptamer candidates and you have one target of interest with 29 interferent molecules, this is going to be 900 combinations that you have to test. If you can do about 4 to 5 ITC in a day, this will take you half of a year of just ITC, which is not ideal. I'm not doing 30 aptamers though. I'm just going to show you guys 4 aptamers that I evaluated the affinity and specificity of with a high throughput method. I'm going to introduce these 4 aptamers. So we have F27, which binds to fentanyl. We have a cocaine binding aptamer, also known as NC48. One that binds to oxycodone, this is OM6, and one that binds to the synthetic cannabinoid AB fubinaca. These have varying secondary structures, but they all bind to their target with nanomolar affinity. I also wanted to evaluate the specificity of these aptamers, particularly other illicit drugs. For example, if I have a heroin aptamer, I wouldn't want it to bind to something like fentanyl. So we want to make sure that it is selective. We also want to check for common cutting agents if we're going to be evaluating what's in a seized sample, as well as drugs that may be found in our everyday body, such as caffeine or maybe something like ibuprofen. So now I want to show you how long it would take if I were to do one of the previously mentioned gold standard methods. If I do 4 aptamers with 24 ligands, this is 96 combinations, which is about 24 days of just characterization. But now I would like to introduce a new high-throughput method that our lab has developed. This is the enzyme digestion assay, and when we use this, I can do all of this characterization just in 1 day. So now I'm going to show you guys the working principle of this new high-throughput assay to speed up aptamer characterization. And this assay utilizes 2 exonucleases. One of them is exonuclease 3. So this is going to digest the aptamer and remove nucleotides from the 3' end of double-stranded DNA. This exonuclease is able to differentiate between unbound and bound aptamers. And I'm going to show you how this works in a few slides. The second exonuclease that we utilize is exonuclease 1, very similar to exo3, except this one digests the 3' end of single-stranded DNA. So this exonuclease can completely digest the aptamer without the presence of target. So what we do is we have our aptamer of interest, and we're going to introduce it to the 2 exonucleases with no target. And what happens is the aptamer is in its unfolded state, so it can be digested by both of these exonucleases, and I'm going to have a bunch of mononucleotides that are left. These mononucleotides, when introduced to a DNA staining dye, will not be stained because they're just mononucleotides, so we'll have no fluorescence. On the other hand, if I take the aptamer and I introduce it to a target of interest, the aptamer will bind to the target, undergoing a conformational change. From there, the exonuclease can only digest a few nucleotides, but it's going to halt digestion near the binding domain. Because of this, we're able to have our aptamer be intact. This can be then stained with a DNA staining dye, such as cyber gold, and we're going to have fluorescence. So no fluorescence means that the aptamer was able to be completely digested. If we see fluorescence, this means that it was able to resist digestion because it bound to a target. This is an example of what our data looks like. You can see that the black line, this is just the aptamer with no target, and over time you can see that it almost reaches 0% fluorescence because it's being digested. On the other hand, if we introduce a target, for example, this binds to acetylfentanyl, we have a very strong resistance value and it is able to resist digestion. We can quantify this by taking the area under the curve of the target divided by the blank and subtract this by 1. So this is a metric that we use. It's called the resistance value. A higher value means it is able to resist more target. Now I'm going to show you guys some data of the affinity and the specificity. So first I want to show the fentanyl binding aptamer. On the left-hand side, you can see that without fentanyl, the aptamer is able to be almost completely digested. As I increase the concentration of fentanyl, I have a direct proportional increase of my fluorescence. So not only can we evaluate the affinity, we can also look at the specificity. You can see that there's very little to no cross-reactivity to other illicit drugs, cutting agents, and other drugs that may be found in the body. This is the oxycodone binding aptamer. You can see again this direct proportional relationship with the fluorescence and target, as well as showing good specificity. This is another example. This is our AB fubinaca binding aptamer. This has moderate binding with slight cross-reactivity to something like diphenhydramine, which is Benadryl. And lastly, this is our cocaine binding aptamer. It has excellent affinity and also has good specificity. So now we want to compare our high-throughput assay to one that's commonly used in the literature. And one that is used in the aptamer field is called the strand displacement assay. And the way that this works is you take your aptamer of interest and you modify it with fluorophore. You then introduce your aptamer to a complementary piece of DNA, and this has a quencher. And what happens is this cDNA will hybridize to your aptamer, bringing the quencher in close proximity to the fluorophore, and it'll quench the fluorescence, so it's turned off. We then introduce the cDNA aptamer complex to a target, and when the target binds, it will displace the cDNA, releasing the quencher near the fluorescence, and we're able to see an increase in fluorescence. So this is an example of a high throughput assay. We can do this, and we can basically do 384 samples at once. Some advantages is it uses broadly available instrumentation, such as a spectrophotometer, and it's also high throughput. However, some disadvantages is that because there sometimes is interaction between the target and the fluorophore, and it can either increase the fluorescence or decrease it. So we sometimes see false results. And also, this assay requires chemical labeling, which just increases the cost. So now I want to show you guys a comparison of our exonuclease digestion assay with the strand displacement assay to see how they compare. First, I want to show you guys an example where the results match each other. So with my oxycodone binding aptamer, you can see that it is specific with the exonuclease assay as well as the strand displacement assay. So these results agree with each other. However, for the majority of the aptamers that we test, this is not the story. For example, with my fentanyl binding aptamer, I do not have any cross-reactivity to chlorpromazine, but with the strand displacement assay, I do. And so we think that this target is interfering in quenching the fluorescence. We then use the gold standard of ITC to confirm if this binding is true, and it was not. Next is our AB Fubinaca aptamer. We did not see any binding whenever we evaluated mephedrone, but the strand displacement assay did. So again, this is another example where it has inconsistent results. We confirmed with ITC that this binding is not true. Lastly is our cocaine binding aptamer. There is no binding to chlorpromazine, but with the strand displacement assay, we see binding. And after confirming with ITC, we know that this is not true. So in summary, our exonuclease digestion assay is a good alternative to a high throughput assay when compared to the more expensive strand displacement assay. And so after we use this characterization technique, we then have an aptamer that can be employed in a biosensor, and we can use this to detect illicit drugs. In our lab, we employ the aptamer-based dye displacement assay. And so what we do is we take our aptamer of interest and we introduce it to a dye molecule. This dye will bind to the aptamer and exist in its monomer form, and it'll have its own optical property. For example, it's pink. But then whenever you add in a target, for example, fentanyl, the fentanyl will bind and will displace the dye into solution. When it's in solution, the dye molecules can aggregate together, and this is going to change the optical properties of the dye, and you can visually see a color change. This assay happens within 10 seconds. You can see a color change. It's quick. It doesn't require any modifications. And now I'm going to show you guys some results. So this is our ultra-sensitive fentanyl color test. You can see that without fentanyl, it's pink. And as we titrate in up to about 2 to 3 micrograms of fentanyl, we can see a color change. More impressively, we can see a color change with 1,000 times fold lower of the lethal dose of fentanyl, which is 2 milligrams. Not only is our assay sensitive, it is also specific. So we evaluated the assay with other counter targets, such as other illicit drugs, over-the-counter drugs, and pharmaceuticals. And you can see it exhibits excellent cross-reactivity. And we also wanted to see, does our assay work for analogs? For example, fentanyl is often modified with various R groups. This test can determine 107 analogs of fentanyl out of 120. And this is just an example showing 40, You can see that out of the 40, only a couple of them do not bind. Not only do we have tests for fentanyl, but we also have tests for other illicit drugs, such as methamphetamine, heroin, oxycodone, cocaine, and MDPV. And this assay is very powerful because we can use different dyes to see different color changes, which is why sometimes maybe it starts off as pink to orange, and others it's green to yellow. So you can optimize this to be whatever color you want. So in summary, our exonuclease digestion is a better approach for high-throughput characterization of aptamer ligand binding, and we can quickly identify a high-quality aptamer that can be used for a biosensor, specifically the dye displacement assay, and for the on-site detection of illicit drugs. And I would like to thank our funding, the NIJ, as well as my advisor, Dr. Zhao, and specifically Gabriel Tin and Dr. Alcamis. They're the ones who helped with the dye displacement part of this presentation. And with that, I would love to take any questions.
We have a microphone coming around. So.
While we're waiting on that, can I ask one of the online questions?
Of course you can.
How was fluorescence measured, and is there an upper and lower detection threshold?
Yes, so we use a spectrophotometer to measure. So for our enzyme digestion assay, we measure between like 495 to 537 nanometers. So that's like the threshold of our of the fluorescence. Yeah.
My question is, how does your research compares to common other high throughput analysis in forensic toxicology like ELISA? Have you maybe in terms of specificity or sensitivity?
Yeah, that's a good question. I personally have not looked at that, but that is something I will look into. Yeah.
All right, we have another online question. Has the assay been tested with drug mixtures as well, or is it only testing for the target compound?
Yes, drug mixtures as well. Not only did we test the counter targets by themselves, but also with, for example, fentanyl.
Any other questions? Oh, we have one up here.
Are you looking at mixtures of purified compounds, or have you extended this to looking at complex matrices in which you're attempting to detect these molecules in those systems?
Yeah, so we have not used the dye displacement assay for something like in vivo.
Yeah.
We would use our other assays, so we have something called electrochemical aptamer-based assays, where we can take the aptamer and employ it in an in vivo sensing mechanism.
Thank you.
If there's no other questions online. Okay.
All right, thank you.
One more question, please.
Oh, hold on. You're not done. Sorry.
In the future, any additional experiments to kind of push your selectivity, for example, with the other synthetic cannabinoids in comparison to the fumigate?
Yes, that's a good question. So currently, we isolated a new THC binding aptamer that can differentiate between other synthetic cannabinoids. Yeah, so we're exploring that right now.
Okay, you can. Sure, go ahead. Sure.
Can you comment on, so let me take a step back. So aptamers typically are notoriously fragile. They can be. Actually, are these, I didn't catch, these are RNA aptamers?
No, ma'am, they're DNA.
They are DNA. Oh, okay, all right. Well, then okay. Then they should be okay.
Yeah, they're stable at room temperature. We do this at,
Okay,
room
all right,
temperature
thank
on
you.
our bench.
I looked at the folding. They were reminiscent of RNA, and so
I see, yeah.
Okay, thank you.
Okay, thank you, guys.
Good.
Okay, thank you.
One more round of applause for Alex. And also, just super quickly, this morning we had about 400 people join us online, so thank you to all of the people who are joining us, and for all of you for joining us in person. Okay, so up next we have Dr. Rabi Musah. She is a professor in the Department of Chemistry at Louisiana State University. Her scientific research heavily leverages the unique capabilities of ambient ionization mass spectrometry for the characterization and identification of forensically relevant complex matrices. And I have had the wonderful pleasure of doing my graduate research with Professor Rabi Musah, and so I'm very excited to see what new novel methods they are working on today. So go ahead, Dr. Musah.
All right, good afternoon. It's great to see you. I'm glad you're here. And I'm going to be speaking with you today about, let me make sure that I get this working. Yeah, I'm going to be explaining, sharing with you today results in my lab, looking at biomarkers, potential biomarkers of cannabis ingestion. This is a challenging problem. It's a non-trivial problem. I'm going to be talking with you today about some of the strides that we've made towards trying to figure this out. And I don't have one of those portable mic things, so I'm going to plant myself here and just use the laser pointer from here. All right? So let's see if this advances. Wonderful. So this work is funded in part by the NIJ, and the opinions I'm going to be expressing are those of myself in the lab and not necessarily those of the NIJ. So by way of overview, I'm going to begin with an introduction, and I'll describe the problem that we're looking at. I'll talk about the objectives of the work, as well as the methods we used, share with you some of the results, and then talk about some conclusions and some future directions. Now, for this crowd, I don't imagine that there's anyone here who doesn't know what cannabis is, although you may or may not have used it. But we will not be discussing those details. But just so we're all on the same page, it's important to note that marijuana is a psychoactive drug that's derived from the leaves, the stems, the flower, buds, and various parts of plants of the genus Cannabis. And most of us know what this looks like. You can spot it. It has a very characteristic appearance, even if you've never really used it before. And the primary psychoactive component is delta-9 tetrahydrocannabinol, commonly known as THC, and its structure is presented down here along with its monoisotopic mass. Because the primary technique that we use for tracking cannabinoids, including cannabis, is mass spectrometry, we'll want to take note of we'll want to take note of the mass, and nominally it's 314, but because the mass spectrometric technique we use ionizes compounds based on pronation, what we will be seeking is M over Z 315 because it's ionized to the protonated precursor. So you know that cannabis is the most widely used psychoactive substance in the world, and this is certainly true also for the United States. And in 2020, there was a survey that was done that showed approximately 33 million people within a 30-day period had taken in cannabis. And we know, of course, also here in the United States that it's increasingly becoming legalized at the state level, and this may have changed a little bit, but you can see that in red are the states where, by and large, cannabis is not legal either on the state level, just generally for recreational use, or for medicinal use. But pretty much everywhere else, there's some permutation on that theme. Now, despite the fact that it's increasingly legalized at the state level, there are some instances where it would be important to know that somebody has consumed cannabis. And one of them would be driving under the influence cases. Another would be where you have a workplace accident where someone has been, for example, operating heavy machinery and is under the influence of cannabis and creates very dangerous workplace conditions. And then increasingly we're finding accidental consumption cases where animals are dying, children are dying, the elderly are exposed to materials that contain cannabis. And so this is an ongoing problem with numerous issues at poison control centers. Typically, when you want to confirm that someone has ingested cannabis, there are invasive sample collection approaches that are followed because you're looking for metabolites of cannabis. And so you usually will typically use blood or urine. You can do immunoassay screenings, and then you can do a confirmatory analysis using some sort of hyphenated technique like GCMS or LCMS, for example. And what's important to note, however, is that these tests confirm ingestion and not necessarily impairment. And so the holy grail is to figure out some way to on the fly, figure out if someone is impaired by cannabis. And so typically when you are testing for cannabis ingestion, one of the key things that you're looking for, one of the key markers would be metabolites, because someone may have handled the material and have THC on their fingers because they've handled the material, but that doesn't necessarily mean that they ingested the material. So there's an important distinction there. So when someone ingests cannabis, of course the body does things to the compound, and you get these THC metabolites. So if we start with the structure of THC, we can first get hydroxylation, where we modify the methyl group up here to introduce a hydroxyl group, and then of course that can subsequently react to give you a conversion to the carboxyl that we have here to give you the 11-nor-carboxy THC. Note that we've got a change in the mass to nominal 344 when we do that. And then, of course, you can convert that into the glucuronide, where you introduce a sugar unit here, and so that gives you another mass of nominal 520. And I'm highlighting these things because we're going to come back and look at them and notice some surprises that we found in the work that we're doing. So the question then becomes, do we have to worry about anything else? And the short answer is yes. It turns out that CBD, or cannabidiol, which is also a prominent cannabinoid, is one that will also have counterparts for the reactions that we just looked at. And so in this particular case, you can have the same sort of conversion to you can get carboxylation out here to give you that. And so that is similar, of course, to what happens with THC. Now, there have been a number of previous studies that have looked at trying to analyze blood, for example, or urine, to figure out if there are compounds present that can be correlated to impairment. And so I just want to cite a few of those here. So Hustis and others used GCMS analysis of blood plasma to try to determine time since last use. And there are challenges with accuracy in some of these cases. Neumeier and others also looked at the ratio of THC to its metabolites, and tried to see if they could couple that to driving impairment. And then Mano and others, and also Hustis and others, looked at recent THC use to try to connect that with creatine. Normalized THC carboxylate in urine. And then people have also looked at non-traditional matrices like hair and oral fluids and what have you. The challenge with these is that the levels are too low. The mass fraction of the relevant compounds in these matrices are too low to be able to get consistent results. And then there's a nonlinear relationship between the amount of cannabinoids that you find in someone's system versus levels of impairment or time since last use. So, as I said, people have looked at all of these. The results, by and large, have been pretty inconsistent, and so basically there are no reliable chemical tests for impairment, again, because of this nonlinear elimination of THC. And there really is not an agreed upon correlation between impairment and various compounds that you detect. And so typically if you stop someone because they seem to be driving while impaired and you do a breathalyzer test for alcohol and they don't seem to be drunk, then there are other things you can do, testing gait and walking and looking at certain features of their eyes. And then if it looks like there are some issues, then you can send them off to a clinical setting so that they can have some testing done, right? So basically that's going to involve the invasive collection of blood or urine, and there's also the challenge of delayed sample collection. So with breathalyzers, you can do the testing on the fly, whereas there's going to be a delay between when you stop the person and decide they can go get this testing for cannabis metabolites in their system, and that delay may also create some challenges. They may be, for example, impaired when you stop them, but less impaired because of the time lag when you finally do the analysis. And also, of course, a clinical setting is required. So we had a hypothesis, which is that there are biomarkers of cannabis use that can be detected in fingerprint residue. And so the idea was to try to test that. And so the objectives of our work were first to determine if there are molecules that are present in the fingerprint residues of people who have used cannabis versus people who have not used cannabis, and then to also determine whether or not the spatial distributions of these molecules will be reflected in the form of the fingerprint ridge pattern, so that you can actually figure out with a simple test that you could potentially do on the fly to see if they have cannabis impairment. And then lastly, we wanted to structurally identify these compounds to see if they could serve as cannabis use biomarkers. And of course, as you can imagine, there's been a lot of work done on cannabis use metabolites, and so we were anticipating that perhaps we might see some of these in fingerprints. And so we wanted to determine if these molecules were there and what they were. And we do this by a high throughput approach. So the idea was to look at the fingerprint residues of many, many different people, and then use chemometric analysis of the results to see if we could tease out certain M/Z values, because we're using mass spec, to determine what these markers might be. So in that regard, it's helpful to visit some of the work that has been done on detection of metabolites in sweat, because it's molecules in sweat that may also appear in fingerprints. And so we have CBD, which has been detected, there's delta-9 THC, which has been detected, and then CBN, which many of you know is a degradation product of THC. And all of these have been actually detected in sweat. So we went into this thinking we might expect to see M/Z values of 315, and maybe for these two, and then 311 for this one, if in fact we could detect these in fingerprint residues. So what's the idea here in terms of how we're going to use this information? Well, the idea is to be able to distinguish between someone who's handled the material only and not ingested it versus someone who's not handled or ingested it versus someone who's ingested it, right? And then the idea is that if these can show up in a fingerprint ridge pattern, or if we take the residues, we can then take the chemical fingerprint signature of the residues for users and non-users. We can apply multivariate statistical analysis to that, and we might be able to get clustering to actually differentiate between the various groups. So that was the approach. And of course, to do this, we needed IRB approval because we're working with human subjects, which we got. And then we did a power test to assess just how large the sample sizes needed to be for our results to have statistical significance. And when we did that, we determined that we would need a minimum of 26 subjects in each one of our test groups. And then we did the subject recruitment. And then the idea here, and you know, the Aptamer talk that we just heard is a nice prelude into this, is that if in fact it's possible to find biomarkers, then we could have someone simply deposit a print, whatever the binding assay is, if it's got a fluorescence readout or a colorimetric readout, then if we simply apply that assay to the fingerprint, it'll light up, and if there are cannabis markers available in the print. All right, and this is something that can be done on the fly. Now, the technique that we used for this work, the mass spectrometric technique, was direct analysis in real time, high-res mass spec. And so what you see is the instrument here, and there's a reason we use this approach. In this instance, it's a soft ionization approach, which means that we get very little fragmentation, which when we look at complex matrices. Now, we do get some in some instances I'm going to show you very shortly, but we get generally very little fragmentation. That's important because it allows us to get the entire chemical fingerprint signature from the complex matrix of the print itself. Then also, there's minimal to no sample prep when your DART ion source is coupled to a high-resolution mass spectrometer. We're using a time-of-flight mass spectrometer. We can get very high accuracy, and the detection limits are very, very good. And it has a number of advantages over GCMS, namely being that you can detect molecules that are not volatile, for example, and you're not inhibited by the molecules that will be picked up by the solvent that is used for the extraction. But more importantly, it allows us to do these analyses in seconds. You can do an analysis of a sample within 3 to 5 seconds, and that enables us to get the hundreds or thousands of replicates that are needed for the statistical analysis to be robust. And so, again, a wide range of compounds we can detect, no solvent dependence, we can look at molecules that are not volatile, and it is extremely rapid. So, now, what did we do for these experiments? Well, it turns out that glass beads are very, very absorbent. And so what we do is we have the subjects simply hold glass beads between their fingers and then rub them for a certain number of seconds. And then we do the same thing for users and non-users, and then we have a blank, which is just glass beads that haven't been handled. And then we proceed with sonicating that in ethyl acetate for 20 minutes and evaporating off the solvent, and then resuspending the residue in 50 microliters of ethyl acetate, and then simply sticking a capillary tube into that mixture and exposing the coated tube to the ion source to the space between the ion source and the mass spectrometer inlet here. And then we analyze the output of that data through chemometric processing. So this is the typical output of that experiment. And so what you see here is a fairly complex mass spectrum. This is what a typical DART mass spectrum of a fingerprint looks like. And this is the solvent at M/Z 89, the protonated ethyl acetate. And this is just a residue from the capillary tubes that we use for the experiment. But you often will detect fatty acids, which are common in fingerprints, squalene, and I've highlighted those here. What you're looking at here are 2 mass spectra rendered in the form of a mirror or a head-to-tail plot. And what you see here is the cannabis fingerprint spectrum from a subject on the top, and one from a subject that had not consumed cannabis on the bottom. And these are the ion counts for each of these are quite different. That's why these look a lot smaller, these peaks, than this one. But by and large, you see many of the same things, because humans have many of the same things in their fingerprints. The bottom line, though, is that it's not possible to simply eyeball these and look to see what the distinguishing features are between users and non-users. So in terms of the treatment of this data, the first thing we needed to do was take the spectra, subtract the spectra of the blank with a Boolean subtraction, and then it is the output of that then we processed using chemometric methods. So in all, we ended up with 136 DART mass spectra for this preliminary study, to which we applied machine learning methods. And just to give you an idea of the kind of output we get from that and what we're looking for, here you can see a kernel discriminant analysis plot. And the bottom line is you're looking for clustering. So in this particular instance, red is the control, and blue would be the people who ingested cannabis within 3 days of the collection of the sample. And the bottom line is what you want to see is clustering. And you can see, so I'm only showing you one rendering of this. If you rotate it around, if you look at it in 3D, you can actually see pretty clear separation between these 2 groups. It looks like there's a little bit of overlap, but it's not as much as is implied by this particular orientation of the plot. But the important thing to note is that you can clearly see that the red samples are separated or clustered together, and the blue samples cluster together. And so the implication of this is that our original hypothesis is indeed correct, or may have some merit to it. And so we have looked at 3 different classification models One of them is kernel discriminant analysis, which was the plot I showed you on the previous slide. Another one is linear discriminant analysis, and then we also did linear regression. In all of these, we can see, using a leave one out cross-validation assessment approach, that we have approximately 86% or higher accuracy in terms of being able to distinguish cannabis user fingerprint residues from non-users. And so we get fairly good accuracies. We're attempting to refine these, but essentially what this is telling us is that the fingerprints of those who have and who have not consumed cannabis can be distinguished from one another. So then the question became, what are the M/Z values that are enabling the models to be able to differentiate between these two classes? And so we set out to determine that, and ultimately narrowed down the list of masses that we got to just those that are relevant for cannabis users specifically. And when we did that, we settled on 14 M/Z values that were of interest, because these seemed to be the ones that the models were using to differentiate between the two classes. So these are the 14 right here, and we are now attempting to figure out what their identities are. But one of the surprises that we discovered was that none of these match the masses that are of known metabolites of cannabis consumption. So in that regard, again, it's important to revisit the sorts of things that one might expect to see based on other works. And so some of the masses of import are the ones that you see listed here. And again, we didn't see any evidence of any of these in the 14 masses that we were looking at. But what we did note is that some of these, like M/Z 81, this is what the formula corresponds to, or M/Z 93 is the formula, this is the formula that, oops, let me go back. Yeah, this is the formula that corresponds to. What we did discover is that those two masses actually appear as fragments under the soft ionization conditions of the DART analysis. And so it turns out that both the terpenes, very common terpenes that you find in cannabis material, as well as several of these cannabinoids, all give you fragments at these two M/Z values. So these results are inconclusive in the sense that while the models seem to need these M/Z values to differentiate between classes, they're common to several of the compounds that we might expect an individual to have ingested. Now, just to show you, this is an example of eucalyptol illustrating the masses at 93 and 81, and then also in THC, you also see these same masses. And so again, this part of it is somewhat inconclusive. So we then moved on to GCMS to figure out whether we could use that as an approach with NIST database matching and what have you to try to figure out what some of these might be. And for the 14, and so we've got a GC protocol that we've developed for this, and what we did was we looked at 5 new cannabis user fingerprint residues and asked the question, do we see any of these 14 masses in these new subjects? And when we did that, what we discovered was that of the 14 M/Z values, one of them consistently appeared, and it was this one at M/Z 299. And so we then set out to try to figure out what that was. And while there's an isopropyl group here on this ester, on this fatty acid ester, we don't really know that it's isopropyl. The work so far seems to indicate that there's this fatty acid that is a good marker for individuals who have ingested cannabis versus those who have not. And this was a surprise. Ultimately, what you really want is a molecule that is clearly derived from the psychoactive substance. And that is not what we found so far. And so we're in the process of trying to figure out what the fatty acid is. It turns out that there are many, many different possibilities here for this, and most of them are not commercially available. So we're doing the synthesis of several of these to try to figure out what that might be. And so with that, we are endeavoring to figure out what those other masses are, and using mass spectrometry imaging, once we figure that out, to see if we can get a fingerprint pattern, an ion image of the fingerprint pattern based on that, so we can differentiate between non-exposure handling and consumption. And so with that, I will conclude by basically indicating that we can differentiate using chemometric methods the mass spectral profiles of users and non-users. There's a subset of M over Z values that is valuable for that, 14 specifically. None of the distinguishing masses or any that we've seen in the literature that serve as metabolites, and some of the detected molecules appear to be fragments of cannabinoids or terpenes. So we're investigating that to figure that out. And so with that, I want to thank the NIJ for its generous support, and also acknowledge Nyara Nichols, who was the graduate student who did all of the experimental work that I've described here. And at this juncture, I want to thank you for your attention. And if there's time, I went over, I will entertain any questions. So thank you very much for your attention.
We do have time for questions, so yes.
Yes, I'm gonna give it to Dr. Frances Scott, because she's a program director, the NIJ.
So is Megan. By the way, we're at 500 total participants online now, so.
Whoa, that's great.
We have two. I'll ask one, and then you can go to one in the room. What are the HRMS parameters, and why wouldn't metabolite fragments be expected in the data?
So what's the first question again?
What are the high-res mass spec parameters?
Yeah, so the instrument is operating within plus or minus 5 millimass units. So we're out to the fourth decimal place on this, operating at 6,000 full width at half mass. And it's not that, so typically under DART HRMS conditions, you're operating with an orifice 1 voltage of 20. And typically under those conditions, you don't see fragmentation. And that's why we like that, because it allows us really to be just seeing the protonated precursors of the molecules of interest and not have the complication of fragmentation. But it turns out that going back, and you certainly know about this from your work, it turns out that those conditions, albeit soft, still cause certain molecules to fragment, and it turns out that both terpenes and cannabinoids fit into that category of molecules that fragment. So we're going to need to get a bit more sophisticated to figure out what those masses are derived from. Yes.
Long nails. Really interesting work. I have two questions for you. Is there a specific reason why you chose cannabinoids apart from it being common? And should I go with the second question?
I can answer this one, and then we'll follow up with the second one. Yeah, so the idea was to try to come up with something akin to a breathalyzer, right, at a traffic stop where, there's a blood alcohol test, and you're actually detecting, the alcohol for the system. And so, and that's how you actually can correlate someone being drunk, right, to, their impairment. And you actually see a number, and that makes it nice and simple and easy. So the idea is cannabis impairment. And so we were specifically interested in cannabinoids because we wanted to know, might it be possible for us to detect a known metabolite in the fingerprint residue in a consistent manner that can be correlated to, ultimately, impairment. So that's why we settled on cannabinoids specifically. And we were primarily interested in THC because that's what the law is built around.
And a follow-up, do you think the same system could then be applied to different classes of drugs?
Absolutely. We can't wait. You taking something we can test? Don't answer that. Yes, it can be, and we have interest in that arena for other types of drugs, yes.
Thank you.
I do. Has this method been tested with other compounds, and it says e.g. syncans, and I'm going to add semi-synthetics, to see if they could produce similar fragments/fatty acids?
It's an excellent question, and the short answer is no. We've not yet extended this to that, but we intend to, but we haven't had a chance yet. Yeah. Thank you.
Hi. You may have answered this already, but how would you distinguish ingestion versus topical use?
Yes, fantastic question. So there are, if a person has handled material, there are cannabinoids that you'd expect to see in their fingerprints just by virtue of handling them, because the body has not had an opportunity to do anything with the compound. So you might see CBD, you might be able to detect THC, but just detection of those by itself does not necessarily mean that the person ingested it. The ingestion would be indicated by the observation of a true metabolite, something that has been modified and is unnatural to the plant material, like the glucuronide, the glycosylated version of the carboxy THC. So that would be the way that we would distinguish between the two. Okay. All right, thank you very much for your attention.
And thank you all for so many wonderful questions for our presenters so far. Up next is Dr. Brent Wilson. He is currently the coordinator of the Cannabis Research Program in the Chemical Sciences Division at the National Institute of Standards and Technology. He is involved with the development of chromatographic methods, primarily focusing on separating many natural and synthetic cannabinoids in complex cannabis matrices. Also had the wonderful opportunity to work with Dr. Brent Wilson. So also just pointing out, this is one of the research projects that we have with our federal partners, so highlighting all the different kinds of research that we fund. So with that, Brent.
Thank you, Megan. That wasn't too bad. All right. Getting it settled?
Working on it.
All right.
While they're working on that, just want to point out that all of these folks were selected to speak before Megan worked here, so this is not nepotism in action. We're just...
Yes. That did cross my mind. This was before she accepted that position. Good? All right. All right. Well, thank you for being here this afternoon. I'll spend the next 20 minutes talking about some of the recent research that was done in our lab. I'm not really going to go into any detail about cannabis, as Dr. Musah said. A lot of you know it already, and she gave a little bit of a background into it. But first, disclaimer, what I say is my opinion, not NIST, not NIJ, and all the blah, blah, blah stuff that's down there. We don't endorse any of the commercial equipment that we will be discussing today during the presentation. So many of you are aware, a lot of considered gold standard is doing gas chromatography with mass spec, LC with mass spec. And NIST, we've done a lot of that work as well, as we've developed methods incorporating those along with LC absorbents. The reason why we do a lot with LC absorbents is because a lot of the forensic labs have gone that way, or at least probably a third of them to do that for quantitation for cannabis. But on the industry side, about 90% of the industry uses LC-UV. So a lot of our work also highlights potential issues forensic labs run into, but also things that could be misleading the industry when they're making their analysis as well. We published some work already on the LC method itself. I'm not going to go into detail with it here. We do have a website that you can find all of our publications on. But what I'm going to focus on today is more work that's actually waiting for review in a journal article now, talking about some of the interferences that we come in to find that could cause issues with LC absorbents and some of the possible solutions that are already being done or could be done. The first interference focuses on CBNA. A lot of you, she brought up before, previous speaker, about CBN being a degradation product. Well, a lot of times people don't talk about one of the precursors of that, which is CBNA. The reason why we found this to be an issue, we developed methods 2020 or evaluated them, hemp samples, everything was perfectly fine. Went to do seed samples, ran into some issues. For example, here are a couple chromatograms from 2 seeds cannabis plant samples from a local forensic lab near NIST. One on the left and right, for the most part, looked like they would be identical. If you just went based off the LC, it looks pretty well. But what actually ended up happening, so if you look at the absorbance spectra, CBN, THCA were fine. But when you went and looked at delta-9 THC, you don't see the same absorbance spectra almost at all. There's definitely something else present there. What that would cause is anybody using LCUV, you would end up reporting the result as inaccurate because you would see that something else was present. It didn't take us long to figure out it was CBNA that was present there based on the retention time and the matching absorbance spectra. Now, the one thing, if you do notice here, most of your instrumentation, you're using PDA, photodiode array detectors. You can get 3D chromatograms. You can see it right off the get-go where the CBNA is present in the 3D chromatogram. So it is something that you should be able to recognize right away. But this also highlights why laboratories, forensics or commercially, need to do additional investigating to make sure that their results they're reporting is actually based on, it is THC as you're looking at. Now, we did confirm this work. So the instrument we have is Symposium LC instrument. It has a deconvolution software on it. And we are actually able to use that to deconvolute. The peak in black is the sample, and then the blue on the left and the red on the right representing THC and CBNA. So we were able to identify using the deconvolution to pull out basically the chromatograms that were for each of those individual ones. So we only looked at 20 samples. And what we actually found was CBNA was in 18 of the 20. We cannot provide accurate results based on that. So actually in the paper, this is exactly how it was reported, detected or non-detected. So one of the things that it led us wondering was, are these just the samples we have? Is it somewhere else? But we wanted to also understand why CBNA was there. So CBN itself is a degradation product that comes from THCA, but as I mentioned, THCA can also oxidize into CBNA, which can then also decarboxylate into CBN. So a lot of times people, when they talk about degradation products, they only focus on CBN. But CBNA is also present there. You can see CBN is very high concentration in these samples, or method quants for CBN. So you can expect CBNA is going to be constantly being developed. So what does that mean? It means you should pay a little bit more attention to your sample storage that you're doing. I would recommend making sure, when we did this analysis, these samples have been stored in the dark for 2 years, 4 years total, but in the dark at room temperature. And then over the time frame, 2 years at the crime lab, 2 years at NIST. So basically, it was a slow degradation of the plant. Now, more than likely, you're not going to have cannabis samples in your lab in storage for 4 years. But it's very possible you could have it for 1 or 2 years, depending on how long it takes for it to work its way through the system. So that's part of what you have to kind of keep in mind as you move forward. Now, these 20 samples we analyzed still had tons of THCA in them, so they were going to continue to produce CBNA and to degrade more to make it the interference still being present there. It's not going to go away. Now, we only did 20 samples, but fortunately in this, we do a lot of work with the industry as well. And I asked the question was, is it just the samples we had, samples that could be degrading in the forensic lab? Well, we found out really quickly, the answer to that question is no. We have a collaboration with Kacha Testing Laboratories. They have 7 different locations. We looked at the data just from one of their labs in Colorado, and roughly 80% of the samples that they tested, which was almost 7,500, had CBNA present in it. So this is not something that's just offshoot that we saw. But the other interesting thing was, I was thinking it was because of degradation issue, the way how we stored our samples. These were all fresh samples that are analyzing for their customers. So what it meant was it wasn't just the degradation product that we're seeing. It's going to be present in fresh samples that you analyze. So if you actually look at it, here on the right I have the graph showing the black is the delta 9 THC concentration in the 7,500 samples, and the red is the CBNA. And what you clearly see, CBNA is present in most of these samples as seen, and as the delta 9 concentrations go up, which also means more than likely the THCA values go up with it as well, CBNA goes up, it goes along with it. So most times when you are going to be analyzing seeds marijuana samples, there's a pretty good probability you're going to be seeing CBA, this interference is going to be present in your analysis. Now, not just looking at the CBNA, when you compare CBN to it as well, it's usually what's found at lower concentrations in the sample, so that shows where you're not seeing the degradation product form yet of CBN because they're fresh samples, and these all had tremendous amount of THCA in it. I think roughly 95% of them were actually above the 0.3% legal limit. So these did have a lot of degradation that would form in the future. So what are possibilities of what you could do to get around this? You could do different chromatographic methods. One of the methods we developed like 6 or 7 years ago looked at using methanol. Our current method from Symposium uses acetonitrile, and that is the primary mobile phase that's used for LC. But when you switched over to methanol, you clearly could see a difference in these separations where delta 9 was separated out from CBNA clearly. The one on the right is actually the method we have that used a 50-50 acetonitrile methanol organic phase for LC mass spec that we developed at NIST. And you can see where delta 9 is clearly separated out from the other cannabinoids there. I should mention, as I will go over in a few slides later, this is just for plants. Finished products will have a different scenario with it. I do get asked by forensic labs all the time, well, can you just change the UV wavelength? This is just overlapping what CBNA and delta 9 look at. You cannot change the wavelengths. No matter what wavelength you're going to use, CBNA is going to have an interference with THC. Mass specs does have the capability of doing it. If you actually look there, they have different precursor ions, they have different transitional ions. Even though this method we have does not have the interference, just to give you an example of how that can work when you have differences, CBC, CBNA do co-elude, but when you do the MRM transitions, you can easily distinguish them from each other. You can do the same thing for delta 9 THC and other components as well. One of the options you could do is deconvolution. I would hesitate you on this possibility because it's only been evaluated with a 1-point calibration curve by the instrument manufacturers. Deconvolution doesn't always work perfectly as it should in the instrumental program, so it is something that you can do. It's good to help verify, but quantitation still needs to be left to be proven. What most forensic labs are doing is actually you don't see CBNA in your samples because you're removing it from the sample. Either by heating it up in the oven to basically decarboxylate any acids that are present, which is nice because then your chromatograms look well. And people are doing this for LC and GC, but it does require additional time. And believe it or not, I've seen a lot of more people's data than most people think, and there are accuracy issues with that. A lot of times labs don't fully validate that procedure like they should. And some labs are just using GC straight up because it converts it all. But there are cons with, there's still a lot of unknown with the decarboxylation efficiency. I will give a little tidbit is look out in the next few months. We are trying to publish some stuff. We've done a lot of work with that with GC. But also a lot of times they do combine that with heating as well. So just for conclusion on the CBN aspect, CBNA interference is these are going to be present in your samples. It doesn't matter if they're fresh or old. So it's going to provide significant limitations on your LC absorbance methods if you're doing that. Make sure, no matter what, you do investigate your chromatographic peaks. This doesn't matter if you're using GCMS as well. We're going to talk about some potential interferences there in a few minutes. So just make sure you kind of keep an eye on that. Storage of your samples needs to be a concern, especially using LC absorbance. I would recommend most labs are taking a portion and grinding it up with a blender or just mortar and pestle. save that portion, stick that portion in your freezer. I'm not saying store whatever 20 pounds you're having in a case. Take whatever you ground up and store that if you're going to rely on LC assorbents just to try and make sure to preserve the sample as long as possible. So the second one that came up with this work was actually surprising because we were only focusing on plant material. But what became popular over the last couple of years is synthetic, well, some people like to call it semi-synthetic THC. It is actually being put in plant samples being derived from CBD. But here's an example of what normal hemp, normal marijuana kind of looks like. You don't see any delta-8 THC in those samples. It lose in this method right before delta-9, or right after delta-9, sorry. But what actually happened is when we actually went and analyzed, these were smokable hemp samples you could buy. You see a lot of delta-8. It was sprayed onto the plant material more than likely. It's basically adulterating the plant from what it was. What we did realize is that we originally thought there was interference coming from the delta-8 peak, but there's actually something we found by looking at the sorbent spectra in between the peaks, that there was actually another component present in there. So when you look at the deconvolution, there actually was another peak found from the delta-8 that's in there. It's a synthetic byproduct. Now, fortunately, this was a very clean sample. I'm going to show you in a minute where they get much less clean. But when we also want to just go back and look at the data we got from the testing laboratory, delta-8 THC was only detected in about 0.5% of the samples. So again, this is not something that's common. Usually this is added to the samples by the manufacturers. Now looking at the 40 samples that actually had it in there, these weren't just hemp samples. They were also adding it to marijuana samples. Do not ask me why. I mean, should already be getting a feel from it, but that's what they were doing. About half of them would have been above the 0.3% limit. So keep in mind that this interference could be in both marijuana and hemp plant samples. Now, why is this a problem? Organic chemistry, you should be able to make delta-8 from CBD fairly easy. Well, the reactions that are used have been around for quite a while, and problem is, though, depending on these certain conditions you produce, you can form many other types of cannabinoids present there. Now, as you can imagine, most of these people are not organic chemists. I am not one myself, and I couldn't explain to you how a lot of these reactions work. But what I can tell you is when you take CBD and you add acid, some type of solvent, and you heat it for at least 60 minutes, you are not going to produce just delta-8 THC. You're going to get delta-9, you're going to get delta-10, you're going to get exo, you're going to get all these other potential cannabinoids that could cause interferences. So this is why a lot of times when you're seeing these type of products, it's not necessarily the delta-8 that's going to cause you issues, it's what else is in there. Now, here are 3 examples of oil samples that we had purchased from some of the finished product work that we're doing. As you can see, in this case here, and this is how most of them look, there's a lot of other components in these chromatograms, not just delta-8 and delta-9. And because the delta-8 is present at such high levels, it also impedes on the delta-9 when comparison. There were a lot of unknowns, unknown 8 and 9. Unknown 8 we could not identify. We bought all the standards, tried to see if it was a cannabinoid. We couldn't. It has a similar structure to some form of THC. For the unknown 9, had a very similar retention time and absorbance spectra, not a perfect match for exo-THC. So I'm not saying it is, but it's somewhere in that family of THC that these synthetic byproducts are showing up from. Now, so what I will also mention here is unknown 10 and unknown 11. I deleted it for time purposes, but those are actually delta-10 THC isomers. So a lot of the work we found when you run into these type of products, you're seeing a lot of the other synthetic isomers in there as well. So just keep that in mind. So what are possible ways to improve your measurements to do this? Similar to what we did with the CBNA, you could change your chromatographic improvements. There on the left, the method we currently use that does separate exo and delta-9. the problem is you can only buy what standards are. A lot of these synthetic byproducts, there are no standards for them. So the really only true way to test your method is to keep evaluating, buy samples, and see if you have any other interferences. Just like the method there on the right was the one we have for LC10 and mass spec I showed you earlier. Well, that was developed for plant. When we went and did it for finished products, because collaborating labs were curious about exo-THC. Exo-THC and delta-9 co-elude completely. And they have almost identical, I mean, you can't distinguish them from mass spec. So that's something to keep in mind is sometimes you're going to have to just choose some limitations you're willing to work with. The important thing is for you to understand and know what those are. The assorbents, you can't use different wavelengths. It's even worse than compared to the CBNA one. All the THCs have almost identical spectra until you get to some of the delta-10 THC isomers. What I would recommend if you're going to go the route of LC is doing some type of combining of chromatographic improvements and MSMS methods. As more and more of these cannabinoids get developed, more and more products get developed, things are just going to get more complicated when you're doing the analysis. So trying to keep that in mind. And here, if you look at, even with delta-9 and delta-8, the precursor and transition ion 1 are the same, but they had different transition ion number 2. So even the MSMS can provide the additional selectivity for you to be able to distinguish some of these. Deconvolution, not even possible. It's too complicated for a lot of these samples. So I would not recommend it. Now, what most of you guys are doing, and actually another option you can do is do GCMS. Now, most labs, this is what you're doing in your lab now. We at NIST developed a method in 2021 for plant samples. We did a little different from most labs. Most labs were doing a 5% phenyl stationary phase. We chose a 35% phenyl just to give us a chance for more selectivity later on because we didn't know what was coming. But we know that now there's a lot of different cannabinoids out there. Before it worked perfectly for the 9 neutrals that were available at the time and also plant samples. So last year we wanted to find out, well, what has changed? Because there's over 40 there's over 40 standards available now. So on the right there, the black is the full scan chromatogram for 28 neutral cannabinoids. We only focused on neutrals because they'll decarboxylate in the inlet. The acids will, so we didn't want to worry with those. A lot more co-elution in that chromatogram, as you can see. So as more standards become available, one of the things we want to know is, one was, could we still use this method for delta-9 THC? But also, we wanted to understand what other interferences are coming up that might cause an issue. Now, in the red is the SIM chromatogram at 299. Delta-9 THC is still, does have partial co-elution with delta-10 isomer, but it's still good enough to use for quantitative analysis. So this was one of the reasons why we chose a 35% phenyl, and it can be extended from plant to finished products. So with that, I'll kind of give a plug for, if you're still here Friday afternoon, this is kind of where I'll pick up from in my presentation in the drugs portion. One of the sessions where we use this method for finished products, oils and vapes, and where you can actually get accurate quantitative measurements. So just in conclusion, for the delta-8 byproducts, hemp and marijuana samples, all of them can be adulterated with synthetic delta-8 THC. You're going to see there are delta-10s out there, even some delta-7. All those are more than likely going to be synthetic additions added to it, so keep that in mind. Sorbents is not going to work very well in a lot of these cases. You should either combine LC with mass spec or do GCMS. Again, as before, make sure you investigate a lot of the potential interferences you can and just understand the weaknesses of your method if you're going to expand beyond what you're currently doing. And with that, I'd like to thank some coworkers at NIST for their work, Aaron Urbis, another colleague of mine, and some students we had there over the summer doing some work, and then Katya Labs for providing some of their data for this, and of course NIJ for funding for this work, and hope to continue that moving forward. And with that, if there is time for any questions, I will take them. There's also, we have a NIST website. that you want any updates, or you can email us at cannabis@nis.gov. I will also throw a plug in there, the bottom right there. Last year, we did release the first hemp reference material from NIS. It's available for purchase if you're a lab that's looking to still validate your method or to continue to evaluate your method. This is a material you can buy. It doesn't have CBNA in it, so it should work really well for what you're looking for. And with that, thank you. Did I talk too long?
No, you're good.
Okay.
Maybe time for one. Go ahead, Francis.
No.
One question online. Each method has limitations, but in your opinion, which method is best to separate the isomers of THC? So start a fight, Brent.
Well, I would say it depends on, based on what I just showed you, I would say it'd probably be GCMS when you start looking at all those isomers. Well, one thing I cannot say is we have not done LC tandem aspect with all the delta 10 isomers. I think there's 4 stereoisomers now, so I can't tell you for certain, but I have a feeling your next presentation might lead into some of that to kind of show where you can use that. So I'll leave that to Rebecca.
Thank you, Brent.
All right.
One more round of applause. All right, so our last presentation for this session is I will introduce Dr. Becky Wagner. She is the Chemistry Research Section Supervisor at the Virginia Department of Forensic Science. Her primary responsibilities include method development and validation for the toxicology, controlled substances, and trace evidence sections. She'll be talking about quantitative method for the analysis of THC isomers, and this is actually one of our publicly funded forensic lab projects. And so with that, go ahead, Becky.
Oh. I don't want to start too soon. We're good? Okay, cool. Hi, everybody. Good afternoon. My name is Becky Wagner, and I'm with the Virginia Department of Forensic Science. Today, I'm going to talk to you about some of our research for the quantitative analysis of tetrahydrocannabinol isomers and biological matrices. So this has been a really great session. I'm excited to present to, especially on cannabinoids. First, before I say anything, these opinions are my own. I am a paid employee of the Virginia Department of Forensic Science, and this work was funded by the NIJ under the publicly funded labs. I'm not going to go into a lot of history with cannabis since we all have had a lot of presentations regarding it, but really we have to recognize that the consumption and utilization of cannabis has been going on for several thousands of years. It's just now been like really morphing into this big bad beast that we all have to deal with, especially within forensic laboratories. The female plants are the ones that we are very familiar with for the drug purposes and drug use, but there are over 500 different compounds in that plant, which is pretty miraculous that it has many different purposes. And there are 104 cannabinoids, which is why all of us talking today have said how challenging this subject is. So we've talked about phytocannabinoids. Those are what are located within the plant. Delta 8 is considered one of those phytocannabinoids, semi-synthetic cannabinoids, as Dr. Wilson was saying, usually in very low concentrations in plant material. But we're going to talk more about toxicology today, so it's important to understand those tetrahydrocannabinol isomers. So there are several isomers that cause us a ton of problems. Delta 8 and Delta 9 are going to be our most abundant, but as Dr. Wilson said, we have 6A10A, we have Delta 10, we have exo-THC, Delta 7 THC, and it just seems like we're always changing and evolving with what isomers might be present. So to really look at toxicology testing, I'd like for all of you to understand that at our laboratory in 2013, we did put a new method online at the time for cannabinoids using LC-MS-MS analysis. It's very different than what our methods look like today because we were only looking for the compounds that we were really anticipating seeing at the time. Delta 9 was only the THC that we were going to be seeing. We didn't need to look at other metabolites. We didn't need to worry about other isomers. Our extraction was a liquid-liquid extraction using 1 ml of biological matrix, which in a lot of ways can be a lot given how much sample we may have for testing. As we move along our journey, we noticed that in casework we were seeing these poor chromatographic peak shapes. On the top here, the kind of poor quality ones, which I apologize, are 2 examples of some of the chromatography that we were starting to see for delta 9 THC. The first one is more of equal concentrations of delta 9 and delta 8 THC. It's kind of looking like that classic split peak that you might see, but it's actually 2 compounds that are co-eluting with one another. And then if there are lower concentrations of delta 8 THC, you start to see a shoulder off of that, the delta 9 THC peak. So looking at it with standards, we noticed that XO-THC, like Dr. Wilson said, perfectly co-elutes with delta 9 THC. What a glorious thing. And delta 9 and delta 8 do produce kind of that split peak look, but we know that that's actually 2 different compounds. So on top of starting to see this weird chromatography and not really being able to deal with it, because in toxicology we report delta 9 THC. If there's another cannabinoid underneath that peak, we are not accurately representing what is in the subject sample. So we started a secondary journey with stop testing limits in toxicology. So our toxicology laboratory is like many toxicology laboratories across the country. We do both antemortem and postmortem analysis. Our antemortem is impaired driving, so we cover all of the impaired driving cases across the Commonwealth of Virginia, which is about 6,000 cases a year. When we do our analyses, we employ, like I mentioned, stop testing limits. So if the blood alcohol concentration is greater than a 0.1% by weight by volume, we would stop testing and move forward with issuing the certificate of analysis. With needing more epidemiological information about what drugs are present and what is the proliferation of drugs, our legislation was really like, hey, what would it take if we did stop testing, we removed your stop testing limits? And it would naturally take a lot. So a baby step into that is that we know cannabis is very prevalent. In Virginia, we have gone through the whole entire process and we are decriminalized and then legalized. So right now, everyone can use it. And everyone does. So we expected to see with the removal of a stop testing limit and testing all cases for cannabinoids, that we would see a 30 to 40% increase in our case volume for our confirmation testing. So we now have to deal with, okay, now we need to deal with the isomers, but we need to make a more efficient method, and we need to make a method that uses less volume. So that's where our publicly funded labs grant comes in. We need something that will drastically help laboratories across the nation with this ever-growing and ever-changing problem. So we wanted to expand the traditional scope of testing to other cannabinoids, just not the traditional delta 9. And then we wanted to investigate various commercially available stationary phases for extraction as opposed to just a liquid-liquid extraction. We have found with our liquid extraction that we have been using or were using since 2013, there's a lot of ionization suppression present with postmortem specimens, and it's highly variable the amount of suppression you would get with postmortem specimens. So we wanted to develop and validate this method in accordance with the ANSI ASB 036 standard practice for method validation in forensic toxicology. So first we talk about expanding our scope of testing. We knew that we were already seeing delta 8 THC, so we wanted to be able to quantitatively evaluate delta 8 THC, and we also wanted to start doing, at minimum, some surveillance qualitatively on some of the other THC isomers that may be present. The difference with working in a publicly funded laboratory is that we have our seized drugs section right next door, and we can see what's going on in seized drugs and what they're getting to then know what we need to start testing for within toxicology. Usually seized drugs starts to see it first, and so we kind of have an insight of what we're seeing locally in our area. This is where it gets fun. We decided that we were not going to continue to chase and evolve our methods like we did for quite some time with NPSs and always having to add and always trying to check. We wanted to create a dual column chromatography system to give us more confirmation that there are not any other isomers present that we know of, and hopefully to identify those isomers that we don't know of at this time. So the thought of this really comes from how we perform blood alcohol analysis. Traditional blood alcohol analysis uses headspace, GC, dual column, FID. Those two columns are of different stationary phases. So you have different retention times of your volatile compounds. The difference is an FID is a non-selective detector, so we're basing off of those differences in retention times to kind of give our specificity. Well, LC-MS-MS, that's a pretty specific technique. We all talk about that all the time, but not necessarily when it comes to isomers, because we need to know and confirm that we're able to separate and distinguish. So we used both a C18 analytical column and a pentafluorophenyl analytical column in our method development, and as you can see, our gradients are very different for both of those methods too. Just to give you your mass spec parameters, one thing to note is that, like Dr. Wilson presented, that there are different transitions that might be possible, but you could also just use the same transitions for all of the isomers. And then so you're collecting just 2 transitions, and that will then decrease your dwell time so you can get a little bit better of an instrumental response for these really low concentrations that we're looking for and not so happy cannabinoids. We're going to take our journey back in time. On the left-hand side is what we saw before with our old method, and this is now the pentafluorophenyl column is what we're now seeing. Delta 8 THC is the one in green. It elutes first. Then we have delta 9 THC and exo-THC. So we do have baseline resolution between those 3 really heartbreakers that are really chromatographically hard to resolve. Once we were happy with that, we were like, okay, maybe this is possible. We wanted to go on to the next challenge, which was the extraction technique. We looked at a variety, solid phase, liquid extractions, protein precipitations, and we landed on a supported liquid extraction procedure. It's a pretty standard procedure, and for the sake of time, I'm not going to tell you all the details about it. But what I will tell you is something else that we looked at that we don't traditionally look at within toxicology testing is the utilization of silanized test tubes. THC has the sticky factor, or what I like to call the sticky factor, and everyone's nodding, so everyone gets the sticky factor. We looked at every step of the process and what the impact of using silanized test tubes or glassware would be for each step of the process. We found that the only significant step in the process was our initial step in aliquoting our biological matrix into the initial test tube, and that was it. So by doing that, it was a little bit of heartburn while we were performing the experiments, but we saved a lot of money on the back end of not either having to silanize tubes ourselves or paying that extra premium for silanized test tubes for every single step in the process. Once we were pretty confident, and everyone knows here that once you start saying, okay, I'm ready for validation, something has to go catastrophically wrong and you have to start over. But we decided to go move forward to method validation following ANSI ASB standard 036, like I mentioned before. These are all of the experiments we performed. That standard has been around for quite some time now, so I'm not going to go into all of the details because I don't have enough time, although I'd love to talk to you all forever about cannabinoids. So we'll first just go over bias and precision. You don't have a method if it's inaccurate. Like, there's no point. So within ASB 036, we evaluate pooled blood samples. So our method was to be validated for pooled blank blood, antemortem blood, and postmortem blood. These are considered 3 different matrix types. You might be sitting there and saying, no, that's just blood. But actually, they're considered in ASB 036 that they are 3 different matrix types. We use our blank blood for our calibrators and our controls within our laboratory. So we needed to not only show that we can be accurate and precise with our measurements in each one of these matrices, but then also our antemortem specimens were still accurate when using a calibration model that is in blood. So we looked at 3 different concentrations, a low, middle, and high, in triplicate over a total of 5 batch analyses, and then we did that additional evaluation to have non-matrix matched calibrators. So here's a table of some of the data that we collected. We have blank blood, antemortem blood, and then postmortem blood at all 3 concentrations going across. And the thing to highlight is that there's nothing highlighted, which means it passed the bias requirements, which is within plus or minus 20%. Cannabidiol and antemortem blood had the highest bias of 15 to 16% across all 3 concentrations, but that's still well within the 20%. So this is a crazy 2 sets of tables, and I don't expect you to memorize numbers or anything, but once again, if anything was out of tolerance, I would have highlighted it in blue. So everything has passed. For on the left, we have within run precision, and on the right, we have between run precision. We have met the percent CV being no greater than 20% for all matrix types at all concentrations. Another thing that I'd like to note is you'll see that on the top of the slide, it keeps saying C18 column. We validated our method quantitatively for both the C18 column and the pentafluorophenyl column, and we wanted to see how they compared to one another to see which one we wanted to use as the primary quantitative column and which column we wanted to use as kind of that like extra spicy check at the end to ensure that we have nothing potentially interfering with our compounds. Ionization suppression we knew was going to be a critical component of our validation. Like I mentioned before, with our previous method from 2013, we knew we saw highly variable postmortem suppression, and we wanted to make sure that we could rectify that. And when I say highly variable, there were definitely at times where it was just completely suppressed and you barely saw anything at all, which is not so great. So we compared post-extraction fortified samples to NEAT standards, is how we perform ionization suppression and enhancement in our laboratory, at 2 different concentrations with duplicate determinations per matrix source. Once again, we evaluated for suppression and enhancement, blank blood, antemortem blood, postmortem blood, and I'm gonna splash a little bit of urine on the slide next for results on urine too. We evaluated 10 total matrices per matrix type. So now here's all our blue, which was not unexpected for us. So the blue indicates that we had significant ionization suppression, which means that we have ionization suppression greater than 25%, or a percent CV of greater than 20%. We were not surprised by this information. But ionization suppression and enhancement on its own is not necessarily a deal breaker with your method. It's not saying, well, now we have to start over. This is really indicating, one, do you have matched deuterated internal standards? Because those will act more similarly to your compounds of interest. And then also, what impact does this ionization suppression have on the other significant components of your validation, like limit of detection and lower limit of quantitation? So for that, when we were looking at sensitivity, we tripled the matrix sources that we evaluated. So instead of looking at 3 matrix sources for the experiments over 3 analytical runs, we looked at a total of 9 matrices per matrix type. In most cases, because we're kind of scraping the barrel of sensitivity plus the ionization suppression, we have an estimated limit of detection that is equal to our lower limit of quantitation. This is except carboxy THC, which has a lower limit of detection at 2.5 nanograms per mil. The lower limits of quantitation are on the screen as well, and I do want to note for our postmortem specimens, we have increased this concentration within casework, specifically for consistency in those postmortem analyses. And another thing to note is that the qualifier ratio for hydroxy-THC, we actually increased that qualifier ratio acceptance criteria from plus or minus 20% to plus or minus 30%. And that's based off of another ASB standard because the ratio is a very small ratio. This slide and the next might be the most important because as Dr. Wilson said, there are so many different cannabinoids, phytocannabinoids, semi-synthetic, synthetic, crazy cannabinoids out there that we wanted to really test the robustness of this method and interferences with whatever we could get our hands on at the time to see if there were any interferences. So because we're doing that dual column system, we wanted to kind of make, it's like a puzzle, really. It was very much like a puzzle. We looked at the pentafluorophenyl column, which is the first data that you see, and then the C18 column after that. With the C18 column, you can see that delta 9 hydroxy-THC, delta 8 hydroxy-THC can potentially interfere with that. But there's no interference identified with the PFP column. So that kind of solves our problem, right? So if we know if we have a delta 8 hydroxy-THC, or we have a peak for delta 9 hydroxy-THC and we don't see a peak in our pentafluorophenyl column, that means that we don't have that there. The only compound that we have found so far that does not resolve from both columns is with delta 8 THC and the 9R delta 7 THC on both columns, it coelutes. So that is the only one that we cannot distinguish between the two columns. And one of the ways to denote this, it's not necessarily a failure of the method, it's a limitation for sure that gets noted in our standard operating procedure and it is recognized on our certificates of analysis. So as we're going along our journey of method development and validation, we started to see several anomalies within our data. And we saw this really small peak noted in the qualifier ion transition of delta 8 THC. It wasn't at the right retention time, but as you can see, it's causing some issues that happen with the integration, and it's just not looking great. We didn't see this at all during method development. So this goes to show that once you get into validation, something has to happen badly. When we started to do this and look at this, we investigated each step of the extraction process to kind of isolate all of these variables, and we found that these were actually coming from the SLE cartridges itself. SLE cartridges can come as naturally occurring or synthetically derived. The naturally occurring is just diatomaceous earth. It's a liquid-liquid extraction with diatomaceous earth as a means to increase your surface area of your biological matrix. So we noted this, but now what do we do with it? It's something in our product. We got lot upon lot upon lot of columns to test the prevalence of this. And sometimes in some lot numbers, it would be 1 in every 10 had an interferent. Other lots, every single one had an interferent. So that's not very great. So what we're working on actually now is our own way to wash the columns and evaluate and validate the integrity of the column after we wash to get this interferent off the column so that we can then perform our quantitative analysis of delta 8 THC. So in summary, we were successful in validating a dual column technique for LCMS analysis. We decreased our sample volume from 1 milliliter to 0.5 milliliters, decreased our sample preparation time, and chromatographically resolve several THC isomers. And I'd just like to acknowledge my department that supports me and all of the crazy research ideas that I have, and specifically our chemistry research section and the National Institute of Justice, publicly funded labs for the funding of this project. And I can take any questions if you guys have it. Do we have time?
You have a question? Okay. We have a question up front. Are there any online, Francis?
Yes. She says
Go
yes.
ahead. You can start with one while we get the mic to Brent.
This is a mic.
It's coming.
Lauren says I can go. If the SLE column is the source of the interferent, wouldn't it be incumbent upon the manufacturer to standardize their process?
That is a lovely suggestion. So we have been working with the manufacturer to kind of standardize their production. We identified the interferon, we collaborated with them and sent them like, hey, this is what it is. We were able to identify the mass spectra of it to try to send and give more information. But we are dealing with diatomaceous earth. So there is only so much consistency you can have with a naturally occurring product. The synthetic products may be more consistent from cartridge to cartridge or lot to lot, but we actually found in our validation or our method development that they did not perform as well as the naturally occurring products.
Thank you for your presentation. Very good. PFP would have been something I wouldn't have expected. Did you choose that from the literature, or did you test multiple columns out?
So the question was, with the PFP column, it's not necessarily the first one that you think of. So how did we come to that conclusion of using that column? We did start with like a tiny little technical note that we found digging deep into the internet from a different manufacturer that was not a pentafluorophenyl column, but claimed to be able to separate out all three compounds. We tried that. It looked really good until it didn't pretty quickly, and the retention time shifted by several minutes, and we were like, what's going on? Our laboratory uses Agilent technologies for most of our instrumentation and our columns, so that's what we know best. And we went with a pentafluorophenyl column as a similar column to that to then be able to develop the method.
We have another online.
Go ahead, Francis.
Which you sort of answered. Has the SLE interferent been identified?
It has been identified. I have no idea what it is. So we've identified that it's coming from the SLE cartridge. The manufacturer doesn't necessarily know what it is, but they know to look for it. One thing that we have done with the manufacturer is given them our exact instrumental parameters, which they have the same instrumentation in their laboratory, so that they could potentially QC check. And then, so what they're doing now is they QC check it and they say, we think we're good here. So they'll send us some that then we can test and we can say yay or nay. So not quite identified, but we do know that just washing the column with a pre-wash consistently gets it off the column.
One more in-person question.
Yeah, and along those lines, hi, Becky, nice work.
Thanks.
That rinsing that you're having to do, is it affecting? Have you had to go back and check on some of your validation to make sure that it hasn't affected the efficacy?
So with the cartridge rinsing, that's one of the things that we have to do. We came up with we're in we think we've got a method phase of life where we've washed the columns because we don't want the integrity of the column to be compromised. And that's the one thing that we don't have guaranteed if we're doing things to that column before analysis. So we've done washing the column and letting it sit for 2 weeks. We've done wash the column kind of sequentially like a solid phase extraction, wash the column, then add your biological matrix. What does that look like? And it seems to be okay, But now we have to go back to our validation and say, okay, what could be affected by this change? The lower limits of detection, the lower limit of quantitation, bias in precision. So there's definitely areas that we have to then reevaluate before we could put that wash procedure online.
Thank you. If we can get a round of applause for all of our speakers. Thank you. All right, so that concludes our Seized Drugs and Toxicology session for today.